In this video, we will discuss the mechanisms of action of antimicrobial drugs, including inhibition of cell wall synthesis, inhibition of protein synthesis, disruption of cytoplasmic membranes, inhibition of metabolic pathways, inhibition of nucleic acid synthesis, and finally, prevention of virus attachment. As we begin our discussion of antimicrobial drugs, we must first understand the concept of selective toxicity. Selective toxicity refers to the ability of a drug to be more toxic to a pathogen than to the host. Selective toxicity is accomplished by targeting cellular structures and metabolic processes that differ in a microbe from the host. To target the microbes selectively, we must know the structural and metabolic makeup of the microbes and be able to compare them to the host cells to find those differences. All of our material covered up to this point has been to learn the structural makeup and metabolic processes of microbes to allow us to gain an understanding of how these drugs work to selectively kill the microbe without harming the host. It is important to recognize that some targets are more selectively toxic than are others. We are less likely to harm the host when we target structures or metabolic processes that are not found on or in the host cell. When the target of the drug is very similar to those of the host, less selective toxicity is accomplished, and therefore the drug is more likely to harm the host. As we have seen this semester, there are a number of differences that exist between prokaryotic bacterial cells and eukaryotic host cells. Therefore, there are a large number of antibacterial drugs. Fungal, protozoan, and helminthic pathogens are all eukaryotic cells. Thus, the search for structural and metabolic differences becomes much more tedious. We also battle viral pathogens with antimicrobial drugs. It is especially challenging to target viruses because they utilize the host metabolic processes. Thus, to alter their metabolism and shut down viral replication means that we must interfere with the host metabolism. Antiviral drugs are very likely to be toxic to the host. Antimicrobial drugs are classified by their mechanism of action. We will study 6 categories of drugs based on the mechanism of action, including inhibition of cell walls, inhibition of ribosomes, disruption of the cytoplasmic membrane, inhibition of the metabolic pathways, inhibition of nucleic acids, and finally, the blocking of attachment of the pathogen to the host. Cell walls are important targets for many antimicrobial drugs. Antimicrobial drugs have been developed against bacterial and fungal cell walls. We will begin with a discussion of those that target bacterial cell walls. As we learned in Chapter 3, most bacterial cells have cell walls that are very important protective structures. The cells act to protect the cell from lysine when exposed to changes in osmotic pressure. Bacterial cell walls are composed of peptidoglycan, a molecule found only in bacteria. Drugs that target the cell walls of bacteria typically react with one or more enzymes needed to synthesize cell wall components. Because they interfere with the synthesis of cell walls, they only effectively target actively growing cells that require new cell wall components. Older, inactive, dormant cells are not affected because they do not synthesize peptidoglycan. As these drugs target peptidoglycan synthesis, their cell walls become weakened and the cells are prone to lysis. Antibacterial drugs that target cell walls may act at several steps to prevent the synthesis of cell walls. We will explore those various mechanisms in another video that surveys some of the more commonly used antimicrobials. Fungal cells also have cell walls that act to protect them against changes in osmotic pressure. Fungal cell walls are composed of a variety of polysaccharides. One particular is the 1,3-D-glucan sugar, which is not found in mammalian cells. A group of drugs known as echinocandins inhibits the synthesis of the 1,3-D-glucan. In doing so, it acts to weaken the cell walls, resulting in lysis of the cell. Protein synthesis is an important feature of all cells. We have seen throughout the semester that proteins act as important components of cells and are also critical for metabolism because they act as enzymes that permit the metabolic pathways to proceed. Protein synthesis is dependent on the action of the cell's ribosomes. Prokaryotic cells and eukaryotic cells have ribosomes that differ in size and structure. As we have already seen, the bacterial ribosomes are smaller 70S ribosomes, composed of a large 50S subunit and a small 30S subunit. The ribosomes found in the cytoplasm and in association with the rough endoplasmic reticulum of eukaryotic cells are larger 80S ribosomes with a 60S large subunit and a 40S small subunit. However, remember that the ribosomes located within the mitochondria of eukaryotic cells are 70S ribosomes and thus are structurally similar to bacterial ribosomes. The difference in the structure of bacterial and eukaryotic ribosomes provide an opportunity to interfere with bacterial protein synthesis, thereby halting their metabolism to kill them. Drugs that target protein synthesis can act in several ways to inhibit protein synthesis. We will explore several ways in which drugs inhibit protein synthesis at the bacterial ribosome in the lecture video surveying common antimicrobials. Drugs that target the 70S ribosome are usually broad-spectrum drugs, meaning that they kill a wide range of bacteria, including gram-positive and gram-negative bacteria. These drugs generally have selective toxicity against bacteria. However, these drugs may also interact with the mitochondrial ribosomes of the eukaryotic cell, since they too are 70S, thereby interfering with their function. All cells have cell membranes composed of a phospholipid bilayer with embedded proteins. As we saw in Unit 1, the cell membrane acts as a selectively permeable barrier to regulate the movement of molecules into and out of the cell. Disruption of the cell membrane's integrity allows the cellular contents to leak out of the cell, thereby killing it. Antimicrobial drugs that target cell membranes usually have specificity for particular microbial groups based on differences in the types of lipids in their cell membranes. Selectivity of these drugs is not exact and therefore may also interfere with animal cell membranes. These drugs are often toxic to human cells. Metabolism refers to all of the chemical reactions of a cell. As we saw in our study of Unit 2, many of the metabolic pathways of cells are very similar in prokaryotic and eukaryotic cells. There are, however, some differences in the enzymes used for these pathways, and some pathways that are unique among some groups of organisms. These differences can be targeted to interfere with metabolism. Drugs that interfere with metabolism are known as antimetabolites. We will examine the specific mechanisms of some antimetabolites in the lecture video in which we survey common antimicrobial drugs. DNA and RNA are critical for the survival of cells. They are necessary to continue to produce proteins required for metabolism. As we saw in Unit 2, the pathways required to synthesize DNA and RNA consist of a number of metabolic steps that each require an enzyme. Interference with any of the enzymes along the pathways can stop subsequent steps. Antimicrobial drugs may act to inhibit nucleic acid synthesis by blocking the synthesis of nucleotides, inhibiting replication, or inhibiting transcription. There are very few differences between prokaryotic and eukaryotic nucleic acid structure or the processes used to synthesize them. Therefore, there are a number of nucleic acid targeting drugs that are limited in their usefulness because they interfere with DNA and RNA of animal cells. In Unit 3, we learned that microorganisms and viruses must first attach to their host before they can cause infection. If we can manage to prevent attachment of the pathogen, we can interfere with the pathogen's ability to cause infection. Drugs known as attachment antagonists are used to interfere with the ability of a virus to attach to the host cells, thereby deterring their ability to cause infection.